right, welcome back everyone from the break. So we are moving into a session where we'll have two featured speakers. So our first speaker is Erik Wright from the University of Pittsburgh. A short digression because one of the themes of this week is nostalgia. Having Erik come and speak is really meaningful to me and to Lauren because he was around when I remember meeting Erik when I started at CHTC in 2014, and when Lauren started in 2013, he was one of the people she met on her first day. So we have known Erik for a long time, and a lot of our work with him has informed how we do facilitation, how we work with people after they graduate, how we introduce people to the OS pool. And so we're really thrilled to personally, I think, to have him here sharing with you today. Yeah, here to move fast and break stuff. So more formally, it's my pleasure to introduce Dr. Erik Wright, who's joining us from the University of Pittsburgh now. He did his undergraduate in electrical engineering and went to work at Apple for a little bit during the years around the time of the first iPad and iPhone releases. And then he went back to school, first for engineering, a master's degree here at UW-Madison, and then ended up staying for a PhD in microbiology, which, so you guess, is a little bit of a switch, so I'm sure he'll share more about kind of that experience, and was one of the first biologists on campus who used the OS pool in a really big way, has gone on to use the OS pool in a dozen different papers, both from when he was at UW-Madison and now at his current position as associate professor at the University of Pittsburgh. Erik's group researches microorganisms and antibiotic resistance as part of the School of Medicine at the University of Pittsburgh. But yeah, his biggest claim to fame is being featured as the main photo, a weekly email sent from the CHTC alongside Lauren. So he's been featured in many of our websites and publications, and it may appear in the talk that he's giving today. So please join me in welcoming Erik back to UW-Madison for his keynote presentation, Biological Discovery at an Unfathomable Scale.
Can you all hear me okay? Is the microphone working? Sounds like it. Okay, good. So it is really an honor to be back here with you all and see campus again. I think this is one of the most collaborative and interdisciplinary groups I've known and worked with and certainly presented to. So I tried to do a good job of making this palatable to everybody, not just biologists. So as Christina mentioned, I am an evolutionary microbiologist. What that means is that I study very, very, very small things that are so small they're near the wavelength of light. But I know many of you are not microbiologists, instead study very, very, very large things. So I thought I'd start with something that connects us all, and that is the unfathomable scale of the science that we're working on and the tools that we're using. So this is a picture of the constellation of Fornax, which you can see from the southern hemisphere. And it's not a particularly famous constellation, but it is the site of one of the greatest experiments of all time. And you may know of this. Oh, let's see if, there we go. You may know of this, but Hubble in 1995 pointed their telescope at a patch of sky that was completely black, representing 1/30 millionth of the sky. And I'm no astronomer, but I'm told that this is one of the most risky experiments there ever was, because they had to look at it for 23 days to acquire an image, and time on Hubble is very expensive. And so they took this risk of acquiring this image and ended up with one of the most iconic images on Earth here, which shows that in this dark spot where almost no stars were known, there were actually 5,500 galaxies. And what that means is if the galaxies are uniformly distributed as we think they are, then there are on the order of 100 billion galaxies in our universe. Truly remarkable discovery. Of course, the James Webb Telescope took a picture in the same place and it has higher resolution. It can see further into the red light spectrum. And so it found even more galaxies, and we now think there's on the order of 1 or more trillion galaxies in the visible universe. James Webb can see all the way back to about 5% of the time since the Big Bang. So some of the galaxies in this image are over 13 billion years old. It's truly an incredible, unfathomable thing. And every time I look up at the sky at night, I think about how much is out there, as I'm sure many of you do as scientists. But I'd like to convince you today that this picture, which is arguably much less remarkable because I took it from my iPhone on a hike, is actually equally unfathomable. Because what it shows is 4 billion years of evolution in parallel across the entire world, coalescing into the lineages that exist now. And it is what Darwin originally described as the entangled bank. That is all the different organisms competing for space and resources and energy in the same place, happening everywhere on Earth. When I was a boy, my parents liked to take me to the Grand Canyon on vacation. And we'd always start off at the Kaibab Plateau on the North Rim, which is the very top there. There it is. You can see it there. And then on 3 separate occasions, we attempted to climb from rim to river and back. We only made it once. It's 14 miles. You have a 1-mile descent down to the Colorado River. And during that time, you actually cross through 500 million years of life forms. The completely unfathomable thing about that is they are all connected to the same universal ancestor. So all of these things, including yourself, are related to each other. And what remains at the very bottom are these fossilized trilobites, which are the oldest macroscopic organisms that we know of. If you go further to the bottom of the Colorado, you're at 1.2 billion years old, and there's these stromatolites, which are the remnants of cyanobacteria mats that existed in the ocean before there were ever macroscopic organisms like the ones that we're used to thinking about. And in Australia, there's actually stromatolites that are 3.5 billion years old approximately, which gives us some indication for how old life is. When we climb back up to the top and we get to Kaibab again, none of us could walk anymore. So in my family, we call it the Kaibab shuffle. And it's basically like this, because you can't walk at all. So it's a famous place in my family. We succeeded one time. It's unfathomable to me, and equally unfathomable with the number of stars, that all of these organisms are connected to each other and also to you. What this shows here is a phylogenetic reconstruction going back on the order of 100 million years of all the different mammals that exist. And we humans are up in the top next to the chimpanzees and then the gorillas. And you go back and back and back to an ancestor that none of us have known. That is many, many, many great ancestors removed from you. And we know that we're related to each other in part because our DNA contains almost the exact same set of functions with many of these organisms, including mice, and has changed over time. But if you compare it, you can very easily see the similarity between our DNA and that of things that we don't think we're related to at all. unless you were to know this. It's a really remarkable discovery in the 1800s that this is actually the case, that we're all related to each other. What's even more amazing is if you go further back. And so we can go back all the way to the last universal common ancestor, and believe it or not, every single organism on Earth shares some genes from the last universal common ancestor, with no exceptions. It's really phenomenal. And so I study microorganisms. A lot of this tree is microorganisms. You couldn't see them without a microscope. Many of them we only know because of DNA surveys of the environment. We actually have never seen them in a laboratory. We don't even know how to make them grow. Approximately 99% of what's out there would not grow in a laboratory without a whole lot of work trying to figure out how to make it grow. Back at the last universal common ancestor, you had a split toward what we now call the bacteria and the archaea. And then the archaea at one point swallowed a bacteria that became your mitochondria and also the chloroplast of plants. And now you have to pay your taxes. We're over here in the Opishkante. They're very difficult to see, but there's a little figurine there of Carl Woese, who was the first discoverer of this actually in the 1970s at the University of Illinois. It's a really, really remarkable thing, and it represents the best of our knowledge of what is life. There's a XKCD I like about this. Your genetic test results are back. Apparently, you're part of an unbroken lineage stretching back billions of years to the early Earth. And we think this is very much true. So this is all very exciting. It's also a genetic risk factor. So that's what I study and why I'm in a school of medicine. So try to understand how evolution shapes not only our genome, but that of microbes and of infections that we may get now. I think that what connects this to the universe, and those of you who study things that are much, much, much bigger, is the scale. So I told you that there are on the order of 1 trillion galaxies. And if we assume that they're like our Milky Way and have 100 billion stars apiece, then there are approximately 10 to the 23rd stars, which is about 30,000-fold the number of grains of sand on Earth. Truly remarkable thing when you think about the entire Sahara Desert and all the grains of sand in it, each one being a star, and then 30,000 stars. And that's just the visible universe. But what's even more big is the scale of the number of cells on Earth, which is believed to be about 10 to the 30th. And then for each cell, there are approximately 10 viruses, particles that are trying to kill it at all times. So there's about 10 to the 31 viruses. Each of those cells contains a genome, and that genome we partition into approximately thousands of genes. In the human genome case, it's about 20,000 genes. Those genes get what we call expressed to become proteins. And because the genes contain the information that gets converted into a protein, the information can be used to make multiple copies of the protein. So the proteins exist on the order of 1 million to 1 billion per cell, depending on the type of cell. For humans, it's on the order of billions of proteins per cell. For bacteria, it's on the order of millions. And so if you think about it, there's actually 10 to the 30th cells times 1 million proteins apiece. So you're at approximately 10 to the 36th million proteins on this Earth at this moment, which is really remarkable when you add in all the diversification through mutation and changes that have happened over time. You have an incredible diversity of different things out there that coexist with each other. Protein's job is to make more cells. So what you're looking at here is a nerve cell communicating through electrical pulses across axons. And what we're going to do is we're going to zoom in on that nerve cell and show you the best depiction of how we think cells work at this moment in time. You're now passing through the outer membrane, through a pore. You just passed through a protein. And now you're inside the cell, and I guarantee you this is very difficult to watch because things are vibrating so fast. At this scale, where each of these proteins, which is a colored blob, is composed of on the order of tens of thousands of atoms, things vibrate very fast, and the world is actually very dense. Unlike our textbooks for high school that you may have seen a cell picture with a bunch of space in it, they're actually chock full of proteins. And there's so many different proteins working together inside a cell. It's very phenomenal. They each have their own function, and they participate in complexes and through biochemical reactions, taking a molecule from one molecule to another, adding little bits to it, doing chemistry, accelerating chemistry that would not occur in reasonable timescales without enzymatic acceleration, to do things like become locomotor proteins that can actually drag things through the cell on filaments. Each of these different colored blobs is a different protein, and they're all working together, and they're all co-evolving within the cell. And so hopefully I've convinced you that the scale of biology is unfathomable, and therefore, in some senses, relatable to the scale of the universe, because both of those things are at unfathomable scales. But if not, maybe this will convince you. So this is how many genes are different in each environment based on our latest information. So simply sample an environment and then cluster the genes into groups, come up with unique genes, and look at how many genes are there. And in some environments, like the built environment, like this one, there's not a whole lot of genes present. And we've more or less sampled it to the point where we've seen them all. Similarly, with the human gut, we've sampled a lot of human guts, and there's on the order of 10 million-ish genes that are unique inside the human gut. But in more complex environments like soil or the ocean, the number of genes, as you can see with these rarefaction curves, it's astronomical. We don't know where they end. It's truly unbelievable. In every single milliliter of ocean water, there are billions of microorganisms, each one most likely different from the other, and each one filled with a completely different assortment of genes. They may share 0 or near 0% of their genes, only those that were passed from the last universal common ancestor, the few genes that we all have. Do they share? The rest might be completely different. So the diversity of genes that is out there is unfathomable. And this is what we know about it. There are approximately 25,000 different genes shown here. This is the metabolism of eukaryotes like us. Most of this is known because of humans and studying mice. Each of these nodes on this graph is a molecule. Each of the genes is a line, and the genes connect the molecules because they do biochemical reactions. Those are broken down into pathways. Those pathways are then colored. And you can see we know a lot about human cells. We know a lot about eukaryotic cells. But actually, we know almost nothing still, because the scale of this pales in comparison to the scale of the number of genes that are out there, the number of different functions that are out there. And so in a typical bacterium, we may be able to assign a reasonable approximation of the function of about 10% of its genes. We might be able to do some sort of assignment to a quarter, maybe even a half, depending on how far you stretch assignment, and saying something like, I think this works on a phosphate. And if that's a good enough function for you, then maybe half of the genes in the cell we can assign a function to. The other half we know 0, absolutely 0. But working on a phosphate is not a whole lot of information. These genes we know a lot about. If we zoom in on this network, this is an example This is the citrate cycle, also known as the TCA cycle. This is how you eat glucose. So if you eat sugar or your children eat sugar and jump around, this is what's going on. It's looping around here, generating lots of ATP, and they're going crazy until it gets slowed down. And this entire metabolic network is packed with these things. What we want to do is we want to be able to take this knowledge and connect in unknown genes. Because the truth is, there's far more genes than we can ever do laboratory experiments. It's just impossible. Laboratory experiments, for those of you who don't go in a wet lab, they're a lot of work. It's pretty manual. It's pretty boring. And there's a lot of things that go wrong, and it takes forever. That's to study one gene. Okay? There's not really a good high throughput way of doing much of this, especially at the level of depth that we'd like to have. And so what we want to be able to do is link in unknown genes into this network without doing a wet lab experiment. We want to be able to do it purely through the dry lab. That's the dream. And to do that, it means we have to use genomes and just genomes. So start from genomes where we know absolutely nothing about any gene, and then use those to say which genes work with a gene that we know something about. If we can do that, then we can transfer knowledge from what we know to where we don't know. That's the dream. And so we'd like to be able to use no prior knowledge to learn about things so that we can spread our information that we do have across more genes. That's the dream of what my lab would like to do. I've been working on this problem for now about 15 years. It's a tough problem. What I can tell you is that it turns out to be as simple as this. What you want to be able to do is say which genes interact with each other and which ones don't. So if you can distinguish the positives, that are the genes that are next to each other in this network, from the negatives, then you've done it and you've solved the problem. That's the goal. This is very hard. The reason why is all of these genes are inside the same cell together, so they all interact. So the challenge is disentangling the coevolution of genes that are next to each other in this network from genes that are a few steps removed in this network. And we do that a variety of different ways using genome information alone. And then we add on top of it experimental information. And then we annotate that with the knowledge that we have. So we can break this down as we would a regular machine learning problem into an ROC curve where we want to have an L where we have a maximum true positive rate at 0% false positive rate. And there's a bunch of different signals that we can pull from genomes to determine which genes are working together, okay? One of those signals you might think of is, well, if genes work together, then they need to be inside the same cell together, right? So they need to be present together. And because all of these different organisms have very, very different gene content, except for those few genes that are conserved across everything, we can use the presence-absence pattern to infer true positives from false positives. And it actually works pretty well. So we can get to about a 0% false positive rate with about 40% true positive rate. Now, one of the challenges of this is that our dream here is to annotate all the genes that are out there. But the problem is you have a multiple testing correction issue, right? So now you have to do all of the genes against all of each other. So you have on the order of, I'll show you later, trillions of tests to do. And a false positive rate of 0 is required to do that, basically. So this isn't good enough, but there's other patterns out there. For example, genes that co-evolve together will tend to co-localize on the genome. There's a bunch of different reasons for this, but you have a genome you can think of if you're a computer scientist as a big string of letters, right? A, C's, T's, and G's for the most part. And the genes are substrings, and those substrings will tend to move near each other through evolution because there's lots of practical reasons why they'd want to be nearby, okay? They're related to toxicity, of having too much of one versus the other. You can regulate them all together. For example, you can turn them all on at once if they're next to each other. So they will tend to move near each other. And indeed, you see that as another signal that we can extract from genomes. So here we have absolutely zero prior knowledge of what each gene does. And yet we can tell you whether they're next to each other in the network with some amount of fidelity just based off of knowing genome sequences. Another thing that you'll observe is that genes that are next to each other will have correlated genealogies. They are passed down through the same cells. They have the same pressures applied to them. So they'll respond with similar rates of evolution that generates correlated phylogenetic trees. And we can use that correlation pattern actually to derive some amount of idea about what genes work most closely with each other. And then the last thing is that some genes that work together create proteins that touch each other. Okay, so they encode for information for proteins that touch. And in doing so, if you mutate one protein at a site where it touches the other also has to mutate. Otherwise, you've maybe broken that interaction. So we can use correlated residue positions. We call these compensatory mutations, where if you mutate one gene, you have to mutate the other. We can look for that signal of compensation, and it gives us some amount of additional resolution. This, unfortunately, is not enough to solve this problem. And so in each of these categories, we actually use 12 different signals that we extract that are mostly orthogonal from each other. And that gives us more fidelity. And then we can combine them all with any sort of algorithm, logistic regression, random forest, whatever have you. And we can do a pretty good job, okay? But it's not perfect. And so the tool that we use for doing this, we call EvoWeaver. We've worked on EvoWeaver for about 5 years. I actually thought of this idea while I was a PhD student here, and it took forever to get it done. At least that's how it feels when you're a new PI. But we did get it done, and now it's published. And this is effectively the state of the art currently for what you can do from de novo to learn about which genes work together. What does that look like in practice? Well, this is a known pathway, okay? So we're taking one molecule called prephenate, and we're converting it into basilicin. This happens to be an antibiotic. So this organism is making this antibiotic from a metabolic precursor through these genes. These genes interconvert the molecules into this final product. And what we'd like to be able to do is infer this network. Now, there's immediately an issue when you look at this, and that is that these genes have an order to them. You go from A to B to G, of course, to F to C to D, which makes a lot of sense, and has a lot more to do with how they were discovered and what order rather than what they actually do in the order of their biochemical reaction. But we have no idea of ordering, right? We only know which ones work together, best case. So our dream here is to be able to figure out which ones work together, and then from that, hope that we can reconstruct this pathway without order. And indeed, this is a pretty typical case. We can do it, but not perfectly. So what we find, and I just made this as simple as possible, without going into the detail, is the green edges we get right. The red edges we miss. The gray edges we get wrong. But what you see is that all of the gray edges don't point outward, they point inward. Meaning that there's no other genes that we think are connected to these things, but we think all these things are connected to each other, which is wrong. The reason why is because this metabolic pathway co-evolves. So all of these different genes are co-evolving inside the cell together, and they show strong signal of that, which indicates to us that they work together. The problem is we can't assign a perfect order to them, but we can pull out these networks. So this works great, at least as well as we can hope for, I think. So the beauty of this, just to summarize, is if I have a gene, I don't know what it does. I can link it in to other genes that I do know what they do. And then we can transfer that knowledge through guilt by association into a network like this. And if you're a biologist and you spend years running experiments in a laboratory, this information is a gold mine for you, because you just took your gene that you know nothing about and you narrowed what it could possibly do from anything to it's probably an X. And that tells you that you should do experiments related to X. And that's super, super helpful. Okay, so that's the biology part. I hope you didn't cringe too much. I'm about to get into the computer science part. So this is probably the part that will interest most people here. So how do we do this and why do we need the grid and why am I here talking to you? So this is actually what the workflow looks like to pull this off. We start off with genomes. We then have to identify the genes. We have to match the genes across organisms. So if I have gene A in organism 1, I have to find gene A in organism 2 if it exists. We then have to take those A to A relations and we have to do it across all the genomes. So we're now going from pairwise to everybody. So we're going to create a group of genes that we call A. We then have to align the genes in each cluster. We have to construct their genealogies, and that goes into EvoWeaver. The output we're currently calling EvoWeb is unpublished, and I'm going to show it to you today for the first time because we're literally there now. And I haven't shared it with anyone before, so we'll see what you all think. This is happening on the grid as we speak. If you do Condor Q on us, this is Aidan Hunter Lachman, myself, and Nicholas Peter Cooley. Here we are. You can see us running our jobs. What you'll immediately notice is that we have a lot of jobs on hold. It's not my fault. And this actually is a really pain in the butt thing about the grid for us is that we keep on going on hold. So we'll go to some site and we'll just hammer it and we'll be put on hold a billion at a time. And then we'll hit our queue for whatever the on hold limit is. I'm sorry if I'm not speaking in Condor terms. Again, microbiologist. That's kind of a lame excuse, isn't it? So we'll go to maximally on hold and then we'll have to release them all. So Nick, who is the brighter of all of us, decided that this was not the way to do things and constructed DAGs to manage DAGs, which is probably the right way. But I don't know. So he has this thing that obscures everything that goes on below. And although he looks like he's doing nothing, he's actually doing more than everybody. He's doing the step that is second here, the matching the genes across genomes. That takes a lot of time. He does it in flights of 50,000 pairwise comparisons at a time. And that's controlled by a DAG. And then we have myself. I do the last step here, which is the last three. And I'll talk about this in a second, but my big one is this last step of EvoWeaver where I do every single pairwise between the gene groups. And you can see I have a lot of jobs here, right? 361,000. That's actually wrong by an order of magnitude, or by 3 orders of magnitude. It's 36 million jobs. When I was a student here, I was interviewed about how this should look because Condor Q was just starting to do this no batch sort of business, or batching rather. And I didn't think to tell them during the interview that you should not truncate at your 100,000th place because some of us like to do millions of jobs. So please fix that. Done is on the order of 10 million. This has been up on the grid a month. Each of those jobs takes about a minute. So although I'm the largest in terms of number of jobs, I'm actually a smaller percentage of the grid time. Eventually, our goal is to manage all of this in a single DAG, and we want to make it loop. So this is kind of a crazy idea. I don't know whether you all support this, but I'm here to throw ideas out there. Sorry, Marone. I'd like to have this live on the grid with like a cron job and just go get more genomes and do more stuff. But we're not there yet. I'll explain to you why a bit. You didn't think I was going to come here with more work for you, did you? Okay, so how does this look from a Condor perspective? So we have to get the genomes from the NCBI at the NIH. That's order of the number of genomes. That goes very quickly, but it has lots of failures of FTP access. So those are little tiny jobs. There's about 5,000 of them because this is our version 1. We're doing 5,000 genomes to start with version 1 of the EvoWeb. We eventually want to do all genomes, which would be on the order of about 2 million currently. So this would go huge. This is actually little. This is why I want to make it live on the grid, because I don't want to have to Condor release my jobs every day. Then we call the genes inside the genome. So we take this big long string and we break it up into little strings. Okay, so we call the genes. That takes a little more RAM, but it's still a small number of jobs. It's order the number of genomes. Then we get into the big step. We have an order, quadratic order step on the number of genomes squared. That takes about 13 million jobs to do this number of genomes. Each one takes about 2 gigabytes of RAM. And for the DAG manager that Nick put together to do this, that takes about a month to do that entire thing for 5,000 by 5,000. I know because we did a pilot with 4,500 and that's approximately where we landed. Okay, so that's 13 million jobs, each one on the order of 1 to 10 minutes. So that's a lot of time. Then we go and we have a problem. What you'll see here is that I have nice Condor terms off to the right, except for this one in the middle, because this is our bottleneck step that I'll get to in a second. We have this major bottleneck that we ran into that we haven't figured out how to do on Condor. And I'm sure someone here who's enterprising enough will tell me a solution to it. But for this small test case, we need 1 terabyte of disk, 10 gigabytes of RAM, and we need 1 job. The problem is it has to run for 14 hours for the small case. Now it's linear time, so eventually we're going to be up at tens of hours instead of 10 hours, maybe even hundreds of hours. Haven't figured out how to put that on the grid yet effectively. And also the disk space, the amount of file transfer for this is enormous.
What are you using for clustering?
I'll explain. You're talking about the middle? I'll explain to you what we're doing in a second. That's our bottleneck for clustering.
Okay.
Yeah, I have slides on that. So thank you, Marone. So then we go back on the grid, and then I have my old-fashioned way of doing things. They try to teach me the new way, and I'm just like, it works for me. So I have 1 batch. It takes about 2 days to create 100,000 alignments of these genes, meaning I take each of those clusters of genes, whatever gene we call gene A, across all the different organisms, and I basically make it into a matrix, a rectangular matrix of characters. So that works pretty efficiently. Then we construct genealogies for all of them. So we create phylogenetic trees. That also works pretty efficiently. I say greater or equal to 1 gigabytes here because we run into memory problems all the time. If we have too much memory, we don't get enough throughput. So we go down in memory, but then we hit memory issues. So we have retries that are if statements that try to bump up the amount of memory over and over again so we don't have to manage it. If that's not the way to do it, you let me know. But so we start off with 1 gigs or 2 gigs, and then we double it, then we double it, then we double it, then we double it. We get fewer and fewer nodes each time, but that's the way that we manage it. So that's the greater than or equal to 1 gigabyte. We have about 100,000 jobs apiece. And then we go into the 36 million, which gets truncated by Condor Q. And now we're at the number of clusters squared. Now you might be thinking to yourself, well, the number of clusters squared, if you have 100,000 squared, that's bigger than 36 million. Yes, that's true. Why are we at 36 million? Because a lot of genes, we can rule out that they work together just based on their presence-absence patterns. If they're never found in genomes together, that's a pretty good indicator they don't work together. So we have some basic filters on that, and we go down from the total number of possible comparisons, which would be on the order of trillion, down to 36 million. That's done in one batch for 33 months. I can tell you, I'm the one who manages that end of things, and I tried running 36 million jobs with Condor Submit over and over and over, and I kept getting errors. And I thought it was because I had 36 million jobs, but actually it was because in my submit file I had new lines. And if you have a new line in your submit file, nope, you're going to get some weird cryptic error message. So eventually I figured that out, and then Condor Q's like ate it all up in 2 seconds. It's no problem at all. Now, I thought this whole time I had to ping Christina and company. So this actually is really easy for Condor to manage, and that's borderline miracle. So thank you all for that. Really, thank you. That made my job really easy because I did not want to have to figure out to do whatever Nick does, because it's just too complicated for me. Let's talk about the bottleneck step, why this is hard. So we've spent the past 2 years working on this bottleneck step. It looks like this. We have 16 million unique genes in this small test case. Okay, so 5,000 total genomes, we get 16 million unique genes. Those are our nodes on this graph. And we have 9 billion pairwise similarities that come back greater than some threshold. Okay, after all versus all comparisons. So 9 billion pairwise similarities, those are edges. So they're weighted. It's a weighted edge graph problem. And we want to convert it into a colored graph. So we want to cluster it and find, this is a problem known in computer science as community detection. I'm going to detect communities on this graph. There's a bunch of lovely ways to do this that occur in linear time. But there's a problem. They occur in linear time if you use this whole graph in memory. Fitting this graph into memory is effectively impossible eventually. Well, I don't have the HPC to do it. It's probably out there, but eventually it would be impossible. So at the end of the day, there's about 100,000 clusters with greater than or equal to 10 genes. We need a certain number of genes to do any comparison. If you have a gene that's just by itself, I can't construct a phylogenetic tree from you. I can't construct an alignment from you. I can't figure out your presence absence. I mean, your presence absence pattern is pretty boring. So we look for at least 10 representatives per cluster in order to move forward. What does our data actually look like? This is an example of the data. So this is the real data. I pulled the head and the tail of it. So you have genome 1291 on string number 1, because some genomes have more than one string. Gene number 38, so we just go left to right on the string, is connected to genome 2444, string 1, gene 3308. And it has a weighted connection of 868.572. I actually did not do the job of truncating this number or rounding it. We actually only store a certain number of digits per number because it saves file space. So we don't need 19 digits of precision. So we narrow everything down to the smallest possible way we can figure out how to do it. In 14 hours, using our tool Exolabel, we now convert this into this output, which contains all of the genes and an assigned cluster number. Okay, so that ends up being 16 million labeled nodes. We do this through a method called fast label propagation. There's a lot of research on this. There's no out-of-memory implementations, so we had to make one, was the challenge. So this thing on the left is on the order currently of about 1/3 of a terabyte. We need to at least use that much space as basically junk files to deal with this. We have to massage it around and do a bunch of stuff with it in order to make this clustering happen in reasonable time. Because when you read off disk, you have much, much slower reads, right? So we need at least this much space, so we end up at about 2/3 of a terabyte currently. And then we, to make it work faster, we use that much space additionally. So we end up at about a terabyte. So a terabyte, we can cluster this graph in 14 hours. How does that compare to other people? So I just over the weekend ran this on our server to just see where we were at relative to all these other algorithms. So we're the purple line. We have a lot of overhead, like 2 seconds of overhead just to run. And then once we're running, we don't overcome our overhead until we hit a much bigger number of nodes. And if you look at this, you'd say, well, you guys put in a lot of work to, to end up very to the slight of the right of the blue curve. You didn't do that great. And I would agree with you based off of this. So our scaling is not unheard of, right? This is a community detection problem that's been studying computer sciences for ages. There's a bazillion papers in obscure journals about this. And we have linear time complexity. So the slope in log space is the time complexity, and the empirical time complexity. And this isn't asymptotic, but it's approximately linear, or at worst case log linear. But there's other algorithms like other forms of clustering that have similarly linear time complexities. What you want to avoid is anything that's super linear by any major factor. But the difference with ours is this: memory consumption. So because we do everything on disk, it actually makes it so we can scale to ridiculously high sizes. This goes out to, I think, 10 to the 8th nodes. And these are synthetic graphs. But I already told you that we're doing this enormous thing with 10 billion edges in 14 hours. So this just continues, and it has actually sublinear scalability. It's like log of n scalability because of the way that we store things. And so you can see that currently using the last few points, the time complexity is the memory complexity is 0.62, so sublinear. And it should stay sublinear far out. But we use a lot of disk, right? So these other algorithms are not using disk. The high throughput computing, high performance computing, the HPC algorithm here is called HIP-MCL. Here it is. And basically they make a sacrifice for the number of cores that they use, which in our case is 1. They use all that are on this server, which is 56, in order to go faster. But because of that, they make a sacrifice of memory. And you can see HIP-MCL, just the memory just balloons. It's not particularly useful. So that's the really remarkable thing, that we've been able to cut down the amount of memory while maintaining the time complexity. So that solves our bottleneck step. So back to where I was before. I told you that we start from genome sequences. We identify the genes, we match them between organisms. That's our quadratic time step. But that can be very easily put on Condor. We then cluster all of those pairwise. That's the graph step that I just told you about. That is our bottleneck, but linear in time. Then we come down here, we have a couple more linear time steps, and we have another quadratic time step. And then I told you we made this thing called the EvoWeb. So, are you ready for the EvoWeb? First ever showing. Unpublished. There's a piece of it. It's actually much, much bigger. But I'm just going to show you little pieces of it. So if you're a biologist, you look at this and you immediately know this to be true. You have the thing that replicates the RNA that makes the proteins. Then you have all the different things that help make the proteins and the things that help to replicate the RNA. Okay, so all of these things are all connected to each other. This is really obvious, well known, and entirely labeled. Why? Because they have nice names. Each of these proteins is very well studied. We have the crystal structure for it, and they're all beautiful. They're amazing. And every single one of these proteins goes back to the origin of life. So all of these are in the last universal common ancestor. We all have them, and so do bacteria. Then you start to get into things we know less about. So this is some sort of genes involved in transposase activity, which is doing something with the DNA to make it simple. And then there's this random ATP binding protein. That's not particularly informative that it binds ATP. Most enzymes bind ATP. It's the currency inside the cell, the energy currency. So the fact that it's linked to these other ones provides an opportunity for knowledge transfer. And you might be looking at the most we've ever known about this protein. But it goes further. Here's a bunch of proteins related to phages. When we know nothing about a protein, instead of calling it an unknown, we call it hypothetical protein, because we're not even sure it's a protein. Most of the time it is, but we call it hypothetical protein because we just have absolutely no name for it. Phage is the biologist term for a virus that infects bacteria. So you can see over here, you have a bunch of phage-related proteins and a terminase involved in stopping replication of the phage. They're all interconnected to all these other hypothetical proteins. So now you've just pulled down a bunch of things from this network that are related to phages. And based on their connections, you can kind of figure out which ones might be doing something or another. Like the fact that one's related to a portal protein might have something to do with the capsid entry, okay, to get the phage to get inside the cell. And it just goes on and on forever. So this is some proteins where some stuff's known about them. Some of them are called domain of unknown function. So we know that there's something there, we just don't know what it does. And then there's the hypothetical protein where we just don't know anything. But they're all related to par A, to which we know a lot. If you're a biologist, and if you study par A, or if you're interested in any of these other ones, this is a gold mine for you, because you've just cut your experimental scope down to a very narrow set of things that you need to study. And when I look at these over and over again, I can't help but start to think that they look a lot like constellations up in the sky. And they're all doing their own beautiful thing together, co-evolving together inside cells for billions of years. And that brings me back to the constellation of Fornax. And so I'd like to say that I think we are all united across all the different domains involved in Contour by unfathomable scales in what we study. And if you heard the title of my talk and heard biological discovery at unfathomable scales, you probably thought that I was talking about how big the OS pool is. But you were wrong, because I would call that very fathomable. In the last year, my group has used approximately 7 lifetimes of computing. I'd consider that fathomable. What I don't consider fathomable is the number of proteins or the number of stars. That's just beyond belief, right? And so all of us across all these different domains are connected by these unfathomable scale of the things that we study. I also think that we are immensely fortunate for the OS pool. And I didn't realize just how fortunate until I recently listened to this podcast. If you haven't heard Co-Recursive, you should go check it out. It's one of those good ones. Co-Recursive podcast episode 109 is really fun. They talk about this game called Tetris. This is Tetris designed to give you the worst block at every move. See how bad it is? This was the world record before they arrived to Tetris. This was a recording of the best gameplay that was ever done by a human. And then people started to use computers. And they eventually solved this problem. I don't know, I shouldn't reveal it for you. Okay, but you could have guessed. They figure out how to do better than the human. I don't want to spoil it. This is really fun. Go listen to Co-Recursive if you don't already. It's definitely for computer scientists. And what I realized when they were talking about going on the Amazon cloud, AWS, for compute time, and how they were talking about having to leverage their mortgages to do this, I exaggerate a little. I realized just how fortunate we are to have the OS pool. That it is just an immense fortune that this exists, that I don't have to worry about the budget associated with paying for this, and that I don't have to worry about telling Marone every day what I'm going to use the OS pool for, right? So it's open compute. That open compute philosophy is a huge deal and separates the OS pool from comparators like the Xseed which has gone on to become something else. I can't remember, Atlas or something. So the OS pool for me is really, really special. Ta-da! World record. When I was growing up, I loved Spider-Man. I am really a nerd, if you haven't figured that out. And this is the first appearance of Spider-Man in any comic. He's famous for saying, with great power comes great responsibility, which is actually a misquote. You can see the actual quote there. With great power, there must also come, hyphen, hyphen, great responsibility, okay? So this was an Amazing Fantasy number 15, which I looked up just to find out. It's $50,000 on eBay if you want to buy it. Or you can get a ripoff for $7. But I think this is true, and I try to emphasize this to all of my students. The OS pool is so big that doing a million jobs can affect the scale of power plants, if you think about it. Doing millions and millions of jobs of this order, I mean, that's a lot of computing to be running all the time. And so I'm a huge contributor to global warming, probably. But I think it's really important that we use it wisely. And I try to emphasize this with all my students over and over and over again, because I see a lot of them go to it and they use it as a crutch. They don't think through what they're doing before they put their jobs on the grid. They just put them on the grid. It's a little bit too addictive like that, you know? So maybe make it harder to do this stuff. That's my other advice. I'd also say that this is a game changer for research. My career has completely shifted trajectories because of this, because I discovered these people and I was in this place at this time. I would not have been able to build all these different tools that biologists use, that our group makes, that I didn't talk about many of them today. But I now have, I counted about a dozen or more papers that have cited the Open Science Grid. And this has, it's not everything I do, but it is a huge fraction of it. And it has changed my trajectory considerably as a researcher. And so for that, I have immense gratitude for everyone that is involved here. And this is the picture that Christina alluded to. Some of you may have gotten this in your inbox before, maybe 1,000 times. Because weekly, I was the poster child for the OSG, come visit the facilitators. I first met Bill Taylor, who I don't believe is here, but he was with the OSG when I started, and this was around 2012, I want to say. And then Lauren, and then it just took off from there. I'd also like to thank all the institutions that have contributed, especially Syracuse University. I'm joking, you're supposed to laugh. Yeah. But really, all the institutions, humongous. University of Pittsburgh's missing here. We have our own supercomputing center, and I have tried to convince them to get involved in Condor. To the degree where I said, okay, I have a 56 core machine in my office. Would you like me to put it on Condor? And I was told that's kind of small for us. It was big for me as an individual. So this is absolutely huge. I'm so thankful for all of you and those of you who work behind the scene to make this actually happen. It's a huge contribution. I also like to thank my group. We are funded by the NIAID currently. If you'd like to know more about us, you can go to wrightlabscience.com. And this is us a year ago at a conference in front of the Pittsburgh Panther. The person who's done, let's see if this is a laser pointer. Okay, great. So the person who did ExoLabel and EvoWeaver is this guy, Aidan Lakshman. This guy, Nick Cooley, is our software engineer. He's a PhD in chemistry and made the switch to the dark side of computing. I did the other way. Started off in computing and made the switch to biology. And he puts a lot of the stuff on the grid. So he's our expert grid user. In fact, he was at this conference a few years ago. Some of you may know Nick. I'd also like to give a shout out, I forgot to say earlier, to those, especially at UW-Madison, who helped make that beautiful JWST picture from the James Webb of the universe. This is a very, very impressive thing to have done. So the astronomy department here participate in that. And lastly, I'll tell you a story. I am from the University of Pittsburgh, but today I'm wearing a University of Wisconsin t-shirt as an alumni. I didn't actually plan that. Yesterday when I was coming here, my bag got lost and ended up in South Dakota. And so when you go shopping, guess what you find? But it's alumni pride. So with that, thank you all. It's really been a pleasure. I hope to meet many of you over the next week, and maybe you can tell me how to do this stuff better on the grid. Thank you.
Perfect. All right. Thanks, Eric. Are there any questions for Eric? Time for, oh, Todd.
Question, comment. Thank you for the awesome talk. I mean, we always talk about in CHTC about how, don't scale your science to the compute, but if you had infinite compute, what could you dream up? And you really embodied that, which is awesome. Looking at and yes, we will fix the Condor Q problem. Maybe
I thought you were going to tell me to move my window, make it bigger.
Yeah, you gotta slide your terminal over.
Exactly.
Looking at the holds, at least yesterday, by far the most common hold was hook prepare job failure. Does that ring a bell with you? Is that what you see? What do you typically see? That's what
the most often one is failure to get from the data directory. So $ data, OSDF, I want to say.
Okay, OSDF.
Failure to capture files from it.
Yep. Okay. We will work on that as well. Thank you.
Question from Taylor Lee on Zoom. Are you currently only using genomes that can be cultured? How do you assemble genomes if they can't be isolated in a plate?
Okay, great. So approximately, as I told you, 1% of life can be cultured. We know this from surveys of microscopy where people have taken environments and looked at how many cells are there and then put them on a petri dish and looked at how many cells are there. And it's about 1%. If you put in a lot of effort, you can culture more of them. By culturing, you have a major advantage because now you've isolated the genome and of one organism because you have a big bunch of it, right, on a petri dish. And now you have only one organism, and that helps you to get the genome. It helps you to assemble the genome as well, which means when you sequence it, you get little tiny strings. They're about nowadays on the order of a couple hundred nucleotides long, so A, C's, T's, and G's. And then you have to use complex algorithms, which increasingly get more complex, to assemble all those into a very big, long string, okay? If you cannot culture, then you have what's called a metagenome, which means you're pulling all the different genomes together. So you have a bunch of them. They're all in the same place together. And you have to use algorithms to disentangle them into individuals. Those individuals are oftentimes contaminated, so they're chimeric. They're composed of multiple different genomes because you just can't disentangle them perfectly. And so we so far have avoided using metagenomes, but when we scale to the scale that we want to, we'll have to incorporate them. There's a lot of issues related to accuracy of genome assembly and completion that we're trying to deal with. Because one of the big problems that we have is if your genome's incomplete, then your presence absence pattern that I talked about is now false. Because you have genes that are absent that are due to incompletion rather than due to the organism actually not having the gene. So right now, what I showed today was uncultured and cultured, but mostly cultured organisms, genomes that are representative of each species. So I didn't talk about that, but there's 4,500, one chosen per species. It's kind of like Noah's Ark. We have our little Noah's Ark of our genomes. We're going to expand out to the other people that are out there and grab more genomes in the future. Yeah, that's the next thing. We're actually starting that on the grid right now. So a little bit of what I showed you is a bit asynchronous because I showed you all the data and I showed you it was computing. Well, we already computed it once before. So the previous time was the 4,500 genomes. We're now moving into a much bigger set and trying to figure out how to get that to work. And that includes expanding beyond our very narrow filtering criteria on the existing genomes.
It was an amazing talk, and I want first of all to thank you for being such a great member of our community for all these years, and all your contribution back to us, including teaching one credit courses in Python in the black and white days. Two comments and then a question. So I don't know whether you're aware that the Condor is used for managing the data coming down from Hubble. So there is an interesting link here between the beginning of your talk and the rest of it. And it also reminds me of Edgar Spalding, who is in botany, that pointed out that the number of seeds of corn planted in the Midwest is about the same order as the number of stars in the Milky Way. And so here you think about the whole plant, and then we can do. Now, you presented a challenge of can we automate the whole thing? Any thoughts of how we can do better in working with a group like yours and prototyping it on your group to address such a challenge? I mean, you know the character, you know what we are doing today. Can we do things different? Can we do better in helping you with the hold? And it's not only here. The thought is asking you what is the main reason for the hold, and trying to solve it because you're a Keynote. So any thoughts or direction, whether now or moving forward, are greatly appreciated.
Yeah, well, thank you for those kind words, first off. Our biggest issue is having the bandwidth to sit down with you all. I think that you're very helpful, and I think that the issue is on our end most.
So if we find a way to reduce the resources needed to, then maybe it will allow us to do better. Our goal is to automate, which means you spend time doing your what I call stinky work, and we do the clean work. But other immediate opportunities for us to reduce the barriers.
So diagnostics would be really helpful. So, and this is a bit off the cuff, but when I use the grid, and I've been using it for a long time now, not relative to you all, but for a biologist, I think I was one of the first major adopters on this campus. One of the challenges is, for example, if I do Condor Q-S, L does it, I get a printout that is enormous, and I can't decipher it. So diagnostics from the user end.
So do you feel that you have a venue to make these statements and say, okay, Christina or Todd or whoever you pick as a victim, that you feel, yes, I can say, I would like to know this. Go ahead and do it. And that is relatively a low threshold. But I want to make sure that it is clear to you and to others that that is acceptable.
Thank you.
And Christina, when Christina starts doing this, I know that we're in trouble.
Hi, Mary Murphy here from UW-Madison. I just had a question about the future of your Evo Weaver. First thing that came to mind was some sort of discoverability, maybe like a publication or somehow, so that you could take a gene and kind of plug it in and then see all the interconnections. I know that's like very simplified. But kind of what is your future vision of that tool, just as a package for researchers to use within their analysis, or something even further than that?
Yeah, so we've thought about that a lot. Biologists largely need GUIs, and because the training's not there. As a community, we desperately need more training. We need more investment in training. We need more user education. I support a code that has on the order of tens of thousands of regular users, and I'm emailed constantly with questions that are fairly straightforward. And I think that reflects just that there's a lot of people entering bioinformatics, and there's not a lot of, even though it's very well documented, there's, in my opinion, I have a conflict of interest there, but I think that there needs to be a lot more training. I've tried to, I tried to do it on this campus. Marone actually supported me in doing this and teaching a class here. And it drew a lot of people. So I taught introduction to programming for scientists, and I got 100 people per semester. And so there's enormous demand. But the reality is most people need a GUI. And so the direction we're headed is toward a website where you shove in your sequence, your A's, C's, T's, and G's, and you get out the network. And then you can basically zoom through it like Star Wars is our dream. We're trying to figure out how to do it on the HTML side is the really tricky thing, because the network's enormous. And whether to do it client side or server side and whatnot, and how to serve that fast enough in a way that's snappy and interactive is really tough. So we've started to put together prototypes for this, but we're nowhere near there yet. And I think it's a future ambitious goal that we might even try to commercialize at some point along the way, because there's a huge amount of demand from industry to understand what proteins do. Because just to take an abstract step back for just a second, AlphaFold was a game changer, because now we can fold any protein into that globular thing that you saw in the video. The problem is that doesn't do a lot for you. So if I fold some hypothetical protein, I get a blob. What I do with that blob is really tough. So the typical thing I can do is I can look for other blobs that look like it. But there's so many different blobs. I know this is like getting very abstract, but there's so many different blobs that your blob almost never will have information known in another blob. So to go further past AlphaFold to the next level is what we're trying to do by connecting proteins to things that don't look like them. So it's not just find something that looks like me, it's find something that doesn't look like me, but I'm still co-evolving with. So that's the dream, and I think there's a lot of opportunity for it. Getting the NIH to buy that is a secondary issue.
All right, let's thank Erik.
Mhm.